And our first speaker is Nisa. So welcome, Nisa.
Hello. Okay. Thank you for the audience that was still stay there. Now, good afternoon. I would like to introduce myself. My name is Nisa Ayunda. I'm from Indonesia, and for now, I'm one of the PhD student at Institute of Oceanography of University of Gdansk. And this time I want to present part of my research in the impact of small-scale fisheries activities towards fisheries sustainability in Indonesia, and supervised by Professor Marius Sapota. Okay. Anyone know where is Indonesia? Okay, so I hope it's a silent question is understand. Okay. Now the title of this conference is where is the world is heading. So for now is the world is heading to Indonesia. No, I'm just a joke. Okay, this time I want to show you a short video about my picture collection of the small-scale fisheries in Indonesia, and I hope it's work. Okay, so. Sorry, the video is silent. Okay, so I hope you enjoy the video, and then so if you are the one interested to the Indonesia, so we are also welcome to go to Indonesia. And according to that video short collection of the picture, small-scale fisheries in Indonesia They usually use small boat. It's the boat that up to 12 meter for length overall, and then some of them use non-machine, and then some of them use the outboard motors. And then they usually use several types of the gill net, and then the several types of the hook and lines. And then some in Indonesia, for the small-scale fisheries, some of them catch the several types of tuna fish, kind of like a skipjack, and then little eastern tuna, and then some of them catch grouper and snapper, and some of them catch the halibut. And then, according to that this condition, this research is our research question is how to govern the fisheries resource sustainability in the small-scale fisheries in Indonesia. And then the purpose of this research is to estimate the rate of the degradation and depreciation through the calculate the coefficient of degradation and depreciation. And then this a little bit simple mathematics. And then this one for the coefficient degradation and depreciation is like a ratio exponent between the sustainable condition with the actual condition. For the example, for the coefficient degradation is the exponent between the sustainable catch with the actual catch. And then in this calculation, we find that the threshold score is mean that in the 0.5, it means that in this condition is the actual condition is higher than the sustainable condition. So if the management of the fisheries activities is better, so it means that this coefficient should be stay away from this threshold score. And then in this research, we find that the biology and economic parameters we constructed by the bioeconomics of Gordon Schaefer and Clark, Yoshimoto, and Poyle models. And then we constructed for fisheries data from Indonesia in 2002 to 2004. 2004 until 2014. And then the results show that actual fishing effort and the production in Indonesia have fluctuations every year. And then this one of the result of the biology and economic parameters you can see in the table. And then for all the data we try to calculate about the rate of degradation and rate of depreciation. For the rate of degradation, we found that the average of the rate of degradation is 0.2, but with the trend increase near to the 0.2 threshold score, and every year. And is for the rate of the depreciation is similar to the rate of degradation. We found that the average score is 0.2, but with the trend increasing near to the threshold score. And this from our study, And research, we can conclusion that small-scale fisheries activities in Indonesia is still causes the increasing of the rate of degradation and depreciation, and it means that the management of the small-scale fisheries activities in Indonesia is still inefficiency. And then from this study, we can recommend it to the Indonesian government that they need to be more stringent in the supervision and monitoring in the small-scale fisheries activities, such as restriction of the fishing effort and increasing cooperation between the local fishermen to develop the national program of conservation. Thank you for your attention.
Thank you. I have a question. Does the analysis of the degradation of these fisheries, does it take into consideration natural disasters or just the local fisheries? As Indonesia, I think, is prone to natural disasters, right? No?
Yeah, from this degradation, because in this research, I want to, we just want to calculation with activities of the fisheries itself, not not with the other impacts of the degradation of the that sometimes the impacts from the disaster too, right? But we just want to calculate and then with how is the real direction for the fisheries activities to the fisheries resources.
Any more questions?
I wanted to ask, is there any sort of do fishermen need to buy licenses in order to fish, or is it just like a free thing to do by whoever wants to?
Yeah, in this situation, we from our national law of the fisheries, from the small scale fisheries, actually they don't need to the license. So, but because of this policies, there's some of the non-government organization asking that it will be caused about the overfishing by itself. So now for around 5 or 10 years lately, our government try to make more data for the small-scale fisheries and then give them a license. But it's still a project in some of the area, not yeah.
Thank you very much.
Okay, so I think it's time to move to our next speaker. Thank you, Nisa. So our next speaker is Karolina Szewc from University of Gdansk, and she will tell us about microplastics in marine environment.
afternoon. My name is Karolina Szewc. I am a PhD student in Marine Chemistry and Environmental Protection Department of the Institute of Oceanography, University of Gdansk. Together with my supervisor, Professor Graca, I am working on microplastics in the marine environment. Today I will describe you microplastic sources, fate and impact on marine organisms. Since 1950, global plastic production has increased from 1.5 to over 300 million tons in 2014. Because of the increasing production of synthetic polymers and their low biodegradability, plastic pollution has become a serious environmental problem. How long does plastic decomposition take? For example, here diapers and plastic bottles, 450 years, fishing lines, 600 years. Plastic cups, 50 years, plastic bags up to 20 years, beverage holders, 400 years, and cigarette butts up to 5 years. My presentation will answer the following questions. What are microplastics? What are the sources of microplastics in the marine environment? What is the fate of microplastics and in this environment, and what is the impact of these particles on marine organisms? Microplastics are particles of synthetic polymers smaller than 5 millimeters, generated by degradation of larger plastic fragments or already produced on millimetric or submillimetric size. In the environment, microplastics are present in form of fibers, irregular fragments, plastic films, or spheres. What are the sources of microplastics in the marine environment? The research conducted, for example, on the Polish coast and the Slovenian Adriatic coast suggests that touristic activity is a very important factor influencing microplastic abundance in beach sediments. So beaches most likely visited by tourists are more polluted with microplastics. The next source are lost or abandoned fishing gear, for example, lines and nets. They are present in seawater and sediments, degrading for many years, mostly by fragmentation. Someone already told us about ghost fishing. So ghost fishing are lost or abandoned fishing gear. Another major source of microplastics in the marine environment is maritime transport. Despite prohibition of plastic disposal at sea, included in MARPOL Convention, scientists still find plastic litter in seawater or sediments. Plastics which originate, of course, from ships. This map here, on this map is marine traffic is illustrated. So as we can see for the Baltic Sea, we can see that the Baltic Sea is heavily exploited by maritime transport. Many research show that microplastics are microplastics which are included in cosmetic and hygienic products that, like facial cleansers, peelings, scrubs and toothpaste, are introduced to the sea with wastewater discharge. these particles from cosmetics, which are called microbats. Wastewater discharge is also a source of synthetic textiles fibers generated during washing. It has been estimated that one washing cycle releases about 2,000 fibers. Can the atmosphere be an important source of microplastics in the marine environment? It probably has a great importance in highly urbanized areas where manufacturing plants, traffic, construction sites, extensive urban infrastructure, and numerous households can emit microplastics into the atmosphere. And these results here show us significant amount of fibers in atmospheric fallout. This leads us to the hypothesis that the atmospheric compartment should not be neglected as a potential source of microplastics in the environment. However, similarly, like as microplastics impact on terrestrial organisms, these issues has been poorly studied. Cliff Coast is a potentially good study area for secondary microplastics pollution due to the protection that it provides from the direct pollution from the land. In our study, polymers present at the cliff coast, here, present at the cliff coast after a storm, has the density lower than seawater and lower than 1 gram per cubic centimeter, and after at a calm sea state, predominant were particles of density greater than 1 gram per cubic centimeter. This indicates that in the coastal zone, plastic debris, especially those of low density, are constantly washed ashore and offshore, which potentially favor their fragmentation. Furthermore, in marine bottom polymers observed in marine bottom sediments had density greater than seawater, so it indicates that they sunk into sediments due to their density. In the study, again conducted in the southern Baltic Sea, most of plastics separated from marine sediments, as I mentioned, had density greater than seawater. So it means they can sink and reach sediments. Because many organisms ingest microplastics, some of them may sink to sediments with fecal pellets, regardless of their density. The presence of less dense particles in sediments is also an evidence of biofouling. Biofouling, it means that after a short time, about 1 week, biofilm develops on litter and its density increases, and before 3 weeks it sinks. As we now know, besides biofouling, also an aggregation with organic matter and subsequent sedimentation could be an important factor affecting the presence of low-density microplastics in marine bottom sediments. Plastic debris in the marine environment contains various hydrophobic pollutants and trace metals and concentrations from nanogram per gram to microgram per gram. Some of these compounds are added during plastic manufacture, while others are absorbed from the surrounding seawater. In general, absorption of organic pollutants increases with time, and when plastic is not aged, like here, and here, when plastic is not aged, the rate of leaching, the release of this pollutants at first increase with time and then decreases. When plastic is not aged, here, this was when plastic is not aged. When plastic is aged, the leaching of pollutants increases with time. So this happens in this way because the leaching of these compounds at first they are released from the surface layer of plastic and then from the deeper parts of this litter. It has been proved that microplastics in the marine environment have an impact on organisms of all trophic levels: worms, fish, sea turtles, birds, mammals. Many organisms confuse microplastics with food or selectively feed on them in place of food. Stomach volume occupied by this debris may limit optimal food intake by intake which is induced by a feeling of satiation, and it may reduce a feeling of hunger, which may reduce the drive to search for food. Subsequently, it may lead to growth rate decrease, decrease of reproductive abilities, the ability to avoid predators. When the gastrointestinal tract becomes completely blocked and severely damaged with abrasions and ulcers by ingested microplastics, it may even lead to death of an organism. In some marine species, organic contaminants, which I said before, can interfere with natural hormone functions, cause mutations and cancer. Thank you very much.
Thank you for the very interesting presentation. We are a little bit short of time, so one quick question, please. So if there is no question, maybe I will ask. What about rivers as a source of microplastics into the sea environment? Are they regarded as a source of the heterogeneous microplastic? Do you know anything about this?
Let's say rivers are not a source of microplastics. They
no,
are
no, as itself, yeah.
yes, itself they are not a source of microplastics, but of course they transport particles from the land to the sea. Also, I didn't mention, but the same we can say about urban runoff. It isn't itself a source, but it transports microplastics to the sea.
Okay, thank you.
Okay, the next talk will be presented by Natalia Sowa.
Good afternoon. My name is Natalia Sowa, and I am PhD student in a department of medical biology and genetics at the University of Gdansk. And the subject of my presentation is the role of the endoplasmic reticulum channels in transport of ricin from the ER to the cytosol. And this subject is just a small part of a big project which is actually realized in our department. So before I begin, let me introduce you plan of the presentation. So on the very beginning in the introduction, I would like to explain what is protein and where are they made, also what is endoplasmatic reticulum and how all of this is connected with the ricin. In the next step, I would like to present the aim of the study, materials and the methods used during my research, results, conclusions and the future directions, and in the end, references. So each cell of human body requires functional protein for its proper functioning. So different cells can synthesize different proteins. It can be, for example, a structured protein. Example may be a collagen, which help to maintain the right structure of the cell. Another example can be a transport protein, which help to move other molecules around our body. And the next example can be enzymes or channels, which is also specific kind of the proteins, which help to communicate the outer world with the inside itself of protein. But let's take a look inside of our cells. So we will find a cell which contains a nucleus, which contain 23 pairs of the chromosomes, which are built of the DNA, double-stranded DNA. So from this DNA, a single strand mRNA is created in a process which is called transcription, and then this mRNA is sent to the cell cytoplasm, and there is folded into amino acid chain, when then it's folded into functional protein, and this process is called translation. So since we know what is protein and where maybe their functions, we also need to know that the process of translation cannot be without irreplaceable machines, which are called ribosomes. It then can be present as a free organelle in the cytosol, or can be bounded with the outer surface or another organelle that you can see over there. It's endoplasmatic reticulum, and their function is biosynthesis of secretory proteins, segregation and export of those proteins to another compartment of the cells, and also folding of the protein. But let me mention that in endoplasmatic reticulum there is also another kind of proteins which are called the molecular chaperones, and they are also responsible for a proper folding of those proteins. So since the protein is properly folded, it may be sent to its place in a cell and perform its function, but not every single protein can be properly folded and then may aggregate, so thus may become a threat to the cell and must be degraded. But the ER doesn't have its own degradation system, so must be sent through the channels which are actually present in the membrane of the ER through the cytosol. And there is a protein complex which is called the proteasome, and that will be cut the small peptides into the small amino acid. And this specific process, which mainly based on the recognition of this substrate and then the retrotranslocation from the cytosol, from the ER to the cytosol, is called the ERAT process. As we know, the ERAT is just a small part of the protein quality process, and it has been demonstrated that the failure of this process can result in many human diseases. So it can be the neurodegenerative diseases like Parkinson, Alzheimer, or Huntington, and then can also be the development of cancer. So currently we know only two types of channel which can be present in the membrane of the ER. So the first one is a protein complex, call it Sec61, Sec from secretory. So the mainly is built from the subunit alpha and with association with two other subunits, beta and the gamma. And we also know that there is another family of proteins called derlin, and they also associate in retrotranslocation from endoplasmatic reticulum to the cytosol. But what is interesting, we still don't know if this translocon Sec61 or the proteins from derlin family can cooperate with each other, or maybe just derlin family can just form a separate channel. So these questions are still without answers. Okay, but let me mention that not only protein use this specific process ERAT to get to the cytosol, but some viruses like HIV or toxin also use this process to get from the endoplasmatic reticulum to the cytosol. But today let me focus only on the one substrate, and that will be ricin, which is built of the two subunits we are connected with each other, and only one subunit, the subunit A, is enzymatically active. So this ricin you can see on the top of the slide is built of the two subunits, and the subunit B, which is marked with a blue color, is bonded to the surface of the cell, and then is retrotranslocated inside the cell through the endosome and Golgi apparatus network. And let me mention that only a small fraction of the ricin is transported in this way. So if we go to the endoplasmatic reticulum, there will be cleavage between these two subunits, and only subunit A through the channels which are present in the endoplasmatic reticulum will be retrotranslocated to the cytosol. So based on the literature, we know that this enzymatically active subunit of the ricin can interact only with the subunit alpha of the Sec61 translocon, and the role of proteins from derli family still remains unknown. What we know about the ricin is that it's the one of the most dangerous toxin in the world. Also cause inhibition of the protein synthesis and cell death. The toxin might be isolated from the seeds from the plants you've seen over there. It's Ricinus communis. And let me also mention that only a small fraction, a couple of seeds, which contain 210 micrograms, is enough to kill a man, which weight is approximately about 80 kilograms. And also it's used as a potential bioterror weapon. So as you can see, the inhaled or injection may cause a very dangerous symptoms and consequently be deadly. So I show you already the bad name of the ricin, but there's also a good one. Ricin can be used in anti-cancer therapy or in development of immunotoxin because this antibody recognize the specific receptors we are present only on the surface, on the tumor cells or cancer cell, and then this toxin, which is connected with this immunotoxin, will be introduced inside the cell and will disrupt the cancer cell from inside. Of course, it's still researches and we still need a lot of information, and also can be used as a vaccines. I'm sure that women should recognize this castor oil, also known as the ricin oil, for amazing properties to make our eyebrow or lashes. But let me also mention that this ricin oil does not contain at all any toxin ricin. So finally, we are aim of the study. So I was trying to check if changes of the amount of translocon protein, which is Sec61 and the derlin, can regulate cytotoxicity and transport of ricin from the ER to the cytosol. As a material, we use a hexalenide, which is widely used in molecular biology. And we use also a technique which is called transfection. So we mix our DNA with a green fluorescent protein, and then we introduce into our cells. So in our case, that was the 3 days, and we were observing the green glowing cells, and that was like a confirmation that our DNA was introduced in the cell. And we make it because we would like to increase or decrease level of these membrane proteins. As a method two, the first one, we were trying to overexpress the proteins which are present in the membrane of the ER. So the first, we were culturing our cells in standard conditions at the media. Then was the transfection with the specific protein, and as I mentioned before. And the next step was the permeabilization with digitoin. Its cells were incubated with a purified ricin, and then the cytosol and the membrane fraction were separated, and then we isolate with specific antibodies only this enzymatically active subunit of ricin. And the next step was detection using a Western blot technique. Another method is a downregulation of the protein, which also were present in the membrane of the ER. As previous, firstly we cultured our cells in vitro. There was a transit transfection, but in this time we used the siRNA, which made that we were silencing our genes, the specific siRNA for a specific gene. And the next step was isolation of the total rRNA, and in the end detection use qPCR. So that's our result, and the result showed the photograph from the Western blot. So as you can see, we successfully separate two fractions, the cytosol and the membrane. And we also identify this enzymatically active subunit with specific antibodies. And we also make like a control, because reticulum, the small protein you can see below, it's only found in the reticulum. So we use a very strong detergent, digitoin, and it was proved that our concentration of this digitoin does not disturb endoplasmatic reticulum, and these proteins were only kept in endoplasmatic reticulum. And the next, there are the picture which shows our result of introduction of siRNA. And as I can see, the smallest concentration of 20 nanomolars cause decrease of level mRNA of a specific subunit alpha SEC61, and that was silencing about 28%. As you can see, as the concentration of siRNA is rising, there's also silencing rising in the cell. But let me also mention that in this case, we also note that when we were silencing this translocon, this one specific subunit of this translocon, we note that the cells are still alive. The viability was not significantly changed. So conclude, overexpressing of DERLIN1 indicates that this protein may be involved in retrotranslocation of the enzymatically active subunit of ricin from the ER to the cytosol. In a condition of decreased level of SEC61 alpha, our results indicate that there must be some alternative transport mechanism. Maybe it's another subunit of the same translocker, or maybe it's just they're in family. We still don't know. And our success was our team optimized condition for transfection with siRNA, which is a very difficult technique. In this case, was only specific for these genes. But in a future direction, we also plan to optimize condition for transfection with combination and separately with protein from DERLIN family. Also, transport in cytosol will be with increased and decreased level on DERLIN proteins, and also we're trying to check and confirm the viability of cells with a biochemical test. So this research contributes to general biological knowledge of human cells and gives possibility to practical use of this knowledge in the future, and maybe to construct therapeutic or antidotum for the ricin which still do not exist. So in the end, let me thank you to my team I'm working with and our department for your precious support, and thank you for your time and attention. Thank you very much.
Thank you. Do you have any questions to Natalia? Okay, so I have one question. Are there other proteins which are transported in cells in the same way as pathways as ricin?
Yes, there's a lot of proteins which use actually error process. They might be course, there might be another proteins which are transported in the same way using ARAT process, because it's, for example, the cholera toxin or Shiga toxin also can be transported this way using the ARAT process or the ARAT system or process.
Thank you, Natalia.
Okay, thank you. And our last speaker is Karolina Pierzynowska, and she will tell us about another approach for treatment of neurodegenerative diseases.
Okay. Hello, my name is Karolina Pierzynowska, and today I would like to present you a novel approach for the treatment of various neurodegenerative diseases. It is known that every genetic disease is rare. However, in highly developed countries, up to 35% of children are admitted to hospitals due to complication of genetic diseases. What's more, there are about 20,000 known genetic diseases. Currently, it is estimated that over 100 million people have genetic diseases worldwide. And finally, at least 70% of genetic diseases affected a central nervous system. According to World Health Organizations, the number of people suffering from neurodegenerative diseases is still growing rapidly, even by 92% in the case of women in the USA during the last 30 years. This is not only a health problem, but also economic issues, because the costs of neurodegenerative diseases in 2010 exceeds $1 billion in the USA and €1 billion in the Europe. As I said before, we have thousands of neurodegenerative diseases. The diagnosis takes sometimes a few years, and finally patients always got an information that there is no cure. So the question is, why is there no cure for almost 20,000 diseases? And the answer is quite simple because pharmaceutical companies invest hundreds of millions of dollars to commercialization one single drug. And these companies compensate for high cost with a large number of patients which are potentially buyers. But in case of rare disease, this high cost of the production of single drug must be compensated with a high cost of single drug. And unfortunately, these medications costs are too high for a single person to pay for them, and the lack of buyers make it unprofitable for a company to produce a medicine for rare diseases. So the second question is how to help patients and to make profits to pharmaceutical companies. And our proposition is to find a common therapeutical targets in various diseases. Therefore, one drug might be effective in treatment of many disorders. So the number of patients suffering in presented diseases considered separately is very low. But if one drug will be effective to treatment them all, the number of patients become as big as number of patients suffering, for example, from asthma. So we started looking for a common feature of all these diseases, and let's look at the most characteristic diseases. It is obvious that they have different causes. Unfortunately, today we can't treat chromosomal aberrations because too many genes are affected in the same time. But from another categories, we choose one representative diseases, and as I said before, they have a different cause, but also it have a different onset and different inheritance. On the other hand, they have one common feature, a disease mechanism, which is a material storage. Although the storage material is different in every of these diseases, we started looking for a mechanism which can remove them all. And we found the process called autophagy. The autophagy begins in cells when the abnormal molecules appears in the cytoplasm. When the autophagy begins, when the isolating membranes encircled abnormal molecules and closed in structures called autophagosome. The autophagosome is then fused with the lysosomes, which are the acid hydrolases with digest functions. The acid hydrolases from the lysosomes digest autophagosome insides. But we must remember that a potentially therapeutic drug should not only induce an autophagy process, but also be safety in long-term use and should crosses the blood-brain barrier, what is the most important thing in case of neurodegenerative diseases. So the compounds which have all these features is genistein. In this is an isoflavones which can be found in vegetables, fruits, tea or wine, and genistein has many biological activities and is widely used in medicine. How genistein, what is the mechanism of genistein action? In normal cells, mTOR kinase, which is a major cellular regulator, keeps a TFEB factor in phosphorylated form. But genistein inhibits an mTOR activity, what results in dephosphorylation of TFEB and its translocation to the nucleus. It results in increased biogenesis of lysosomes and increased activity of lysosomal enzymes, what leads to increased efficiency of autophagy process. What's more, genistein crosses a blood-brain barrier and is safe in long-term use, even in the high doses. So let's back to our three chosen diseases. The first disease is what was, which the genistein was tested for, was mucopolysaccharidosis. Mucopolysaccharidosis are caused by accumulation of glycosaminoglycans or GAGs, and in normal cells, GAGs are degraded from cells by many of enzymes in reaction, which is presented here. But the lack of any enzymes leads to the accumulation of glycosaminoglycans in cells and leads to the reveals a lot of symptoms which are listed here and always lead to death before maturity. So the first experiments was performed in vitro, and our results showed that adding genistein to the cell culture decreases level of glycosaminoglycans in cells. The next experiments was performed on the mouse model of mucopolysaccharidosis, and these results also show a decreases level of glycosaminoglycans in the tissue of these animals. And crucial experiments with a behavior was performed. As you can see, mucopolysaccharidosis mice reveals hyperactivity, which we can look here, and animals treated with genistein behave exactly like healthy animals. So the second disease which genistein was tested for was Huntington disease, and our results show that Huntington disease is caused by a mutation in HTT gene, which is located on chromosome 4, and this mutation occurs as an expansion of CAG repeats in the first exon of gene. In cells of healthy people, the number of CAG repeats are up to 26, but Huntington disease is diagnosed when number of CAG repeats exceeds 40, and increased number of CAG repeats results in extended polyglutamine region in Huntington protein, and this mutant protein tends to form aggregates in neurons, what impairs their function and leads to the severe symptoms, of which the most characteristic are sudden uncontrolled movements. So our results show that genistein significantly decreases not only the level of mutant huntingtin aggregates, but also its soluble form. Also, the number and size of huntingtin aggregates was significantly decreased after genistein treatment, and these decreases of mutant huntingtin was correlated with the increases viability of cells, and these promising results led us to planning experiments with animal model of Huntington disease at the end of 2018. And the last disease what was tested was Alzheimer disease. Alzheimer is caused by paired helical filaments consist of hyperphosphorylated tau protein and senile plaques, which are consist in beta amyloid, and this structure impairs of neuron function and leads to the reveals of severe symptoms, of which most characteristic are progressive memory disorders. So we work with a mouse model of rat model of Alzheimer disease, and we divide animals into two groups, a control group and a group treated with genistein for 1 month, and then a memory was tested with in a Morris water maze. Morris water maze is a pool which is divided into 4 quadrants. In one quadrant we put a platform, and the rat wants to get an escape from the water, find a platform. And healthy rats can find the platform quicker and quicker every day, but neurodegenerative rats find the platform only randomly with every follow day. So I want to show you an experiment. Sorry, technical problems. Okay, here you can see a rat treated with genistein, and as you look, he found a platform right now. But the rat from the control group is still looking for the platform. Yeah. And this is the last day of the experiments. So finally, in conclusions, genistein can improve various neurodegenerative diseases, and it can be considered as a common drug for this group of disorders. Thank you for your attention.
Thank you for the presentation. Are there any questions? We have some time for them. Yes.
Okay, I'd like to know how was the genistein administered?
Oral.
Oral. Okay, thanks.
Hi, I'm curious, how do you get the rats to be sick? I mean, you don't really find all the rats with Alzheimer's disease on the street, but do you induce it somehow? What's the method here?
Yes, we use the streptozotocin. It's a toxin, and injection to the brain in rats of streptozotocin induced on Alzheimer disease, because the beta amyloid and senile plaques and hyperphosphorylated form of tau protein is accumulated in the brain. Yeah.
Okay, thank you for the presentation.
So we're going into the poster session now, so please stay by your posters. And this will be the final session of this meeting. I would like to, I didn't say it at the beginning, but I guess it was in our announcement. We offer three prizes: one for the natural sciences oral prize, for humanities and social sciences, and for the best poster without the distinction between the scientific fields. So the next stage will be the posters, then we go and discuss and then we have the announcement, so please stay with us till the very end. You never know. So, the poster session, right? Okay, here we go.